Now I'm going to wrap up chapter 24 on our digestive system. All right, now moving into our small intestine. So we talked about the pancreas and the liver, stomach, small intestine. Now moving into our large intestine. This goes from that ileocecal junction that comes in from the small intestine all the way through the anus. We have three parts: the cecum, the colon, the rectum, and then it ends in that anal canal. So the movements here are sluggish. It takes much longer, 18 to 24 hours compared to like around 4 or 5 hours in the small intestine. So the majority of time is spent actually in the large intestine, and that conversion takes the chyme and converts it into feces. During this process, water gets absorbed as well as some salts. We get secretion of more mucus, and we have different microorganisms. So what's referred to as our gut microbiome, which includes all the different bacteria. The majority of the microbes in our body live in the large intestine. So there are a lot of actions happening, some fermentation, some breakdown of food by these microorganisms. And then just to kind of put it in perspective, again, not numbers you would be required to know, but about 1,500 milliliters of chyme enter the cecum, and about 90% of that is reabsorbed, so that goes from 1,500 down to about 100, so between 80 and 150 milliliters of feces. So a lot of reabsorption happening in the large intestine. So looking at the parts here, so that cecum, again, this is where it's entering from the small intestine, which is just kind of a blind sac. We have what's called the vermiform appendix that's attached there. And those walls contain various lymph nodules that help, again, with our immune system and the absorption of fat. Then the colon, so we have a few different parts here of the colon. So we have the ascending, transverse, descending, and sigmoid regions of the colon. So these have a circular layer with an incomplete longitudinal layer. So the contractions of tennae form these pouches called haustra. And then those pouches are called omental appendages that attach to the outer surface. So it kind of looks bumpy almost. And then the mucosa is made up of simple columnar epithelium, lots of glands called crypts, goblet cells, again, predominate. We have a lot of mucus secretion, but there are also some absorptive and granular cells that we had similar to the small intestine. Then moving into the rectum, just a straight muscular tube, and then into the anal canal, which is just a few centimeters of the digestive tract, made up of superior epithelial of simple columnar cells. Then the inferior epithelia has stratified squamous. And then we have two sphincters here, the internal and external. The internal made up of smooth muscle, external of skeletal muscle. And then the formation of hemorrhoids are whenever the veins enlarge or become inflamed and lead to that painful production of hemorrhoids. Some of the secretions that occur in the large intestines, we already mentioned, again, a lot of secretion of mucus that happens here to provide that protection. Again, digestion's parasympathetic, that increases that secretion of the mucus from the goblet cells. Again, that normal microbiota, lots of microbes live in our large intestine. They're making different acids and some antiporters that help remove acid that line the large intestine. So we get the exchange here of bicarbonate for chloride ions to help with that neutralization, as well as hydrogen ions for hydrogen, sodium ions for hydrogen ions. So the combination of those two exchangers help decrease the acidic nature inside the large intestine. And then water leaves the lumen by osmosis, so the diffusion of water, which is controlled though by the concentration of solutes like sodium and chlorine. So as they move into the epithelial cells, water goes with them, and that allows that reabsorption of water. And then the microbiota produces different gases that leads to flatulence as they break down and ferment various carbohydrates. They those bacteria, though, not only help with the digestion, but also form vitamin K that then gets absorbed. And ultimately, the feces consist of water, and then anything undigested, like cellulose that makes up the plant cell walls, microbes are going to go out through there, as well as any epithelial cells that were lost during the process. So the movements here, we talked about peristalsis. In the large intestine, though, the movement is called mass movements. This is common, especially after meals. That's part of our enteric nervous system reflex, again, that local control. So we have gastrocolic. This is initiated by stomach distension that's going to trigger these mass movements. And then the duodenocolic, which is distension of the duodenum, that's going to trigger these mass movements. Again, mediated by parasympathetic reflexes as well as local reflexes, and then some hormones, CCK and gastrin, as well as even the smell of food can trigger these mass movements. So then the process of defecation is the contractions move the feces toward the anus, kind of in this coordinated relaxation of those sphincters. Then the parasympathetic reflexes allow for most of the trigger for defecation. There's also conscious control of that defecation reflex. So what's happening here are those action potentials from the sacral spinal cord go to the brain and parts of the brainstem and hypothalamus that inhibit or facilitate that reflex activity. Those action potentials also get propagated to the cerebrum, where the awareness of the need to defecate is realized, so that kind of urge to go. And then the external anal sphincter has skeletal muscle, so it's under conscious cerebral control. So if it's relaxed voluntarily, feces are expelled. However, increased contraction prevents that defecation, and then that defecation reflex persists for only a few minutes and then quickly declines. So it can also be initiated by voluntary, what's called the Valsalva maneuvers. So this straining includes a large inspiration of air, followed by the closure of the larynx, and then a forceful contraction of those abdominal muscles. And then increased intra-abdominal pressure forces the feces to the rectum, and then that stretching initiates the defecation reflex. All right, so now if we think about, again, now that we've moved through all the parts of the digestive system and accessory organs, now if we actually talk about the digestion and absorption of these nutrients. So digestion is just a breakdown of the food for absorption. So again, mechanical, which is kind of the chewing, breaking those large food particles into smaller ones. And then the chemical digestion, which are going to be the enzymes breaking down the bonds that hold those molecules together. Absorption and transport, so as the molecules move out of the digestive tract into the circulatory, that's what we're referring to as absorption, and then distributing through the body, through the cardiovascular system or lymph, would be the transport. So some are absorbed by diffusion, just going from high to low concentrations, but the majority of them need transport proteins because they can't pass through that phospholipid bilayer of our membranes. So we have a couple sides here. So the side of the epithelial facing the lumen, the internal part of the digestive system, that's called the apical membrane. And then the part that faces the blood vessels is called the basolateral membrane. So apical is facing into the digestive tract, basolateral toward the blood vessel. So then the transport to water ions and these water-soluble products like glucose, they enter the hepatic portal system, and then that transports them to the liver and then ultimately into the cardiovascular system. The products from lipid metalysis, though, because lipids don't like water, they're coated with proteins and transported more into the lymph capillaries that are called lacteals. And then they connect those lymphatic vessels to the thoracic duct and then ultimately into the cardiovascular system. And those proteins that coat the lipids, those products, whenever they travel to the blood, then they go to the adipose tissue or to the liver. So if we look at the three main types of digestion, so whenever our food comes in, they mainly comprise of carbohydrates, lipids, and protein. So the carbohydrates are long chains of like glucose. So those are broken down into monosaccharides. Lipids like triglycerides, they're broken down into fatty acids, and monoglycerides, which have this glycerol molecule in just one fatty acid. And then amino acids or proteins are broken down into amino acids. That's the digestion that we're discussing. So if we look at carbohydrates, so we have polysaccharides, which are chains of lots and lots and lots of monosaccharides, like cellulose starch. Disaccharides are two, so like lactose or table sugar, which is sucrose. And then individual sugar units like glucose or monosaccharides. So as we mentioned before, we get some digestion in the oral cavity from salivary amylase. That gets inactivated though in the stomach. So then whenever we move to the small intestine, that pancreatic amylase and the disaccharadases get released to break down the polysaccharides and the disaccharides into glucose and other, and then some disaccharides. So then what gets absorbed is glucose can move through a glucose transporter. Galactose can also be taken in by using what's called a symport. So sodium moves with its concentration gradient that gives energy to actively move galactose. And then fructose can also be absorbed by facilitated diffusion, just meaning using a transport protein moving from high to low concentrations. If we look using glucose as an example here of how it moves across the epithelial tissue. So again, here's our lumen side. So this is that apical membrane. So again, we have glucose hanging out here in our digestive system. This is using a symport. So glucose is coming in. It needs energy though to come in. So that energy is provided by a symport. So sodium is coming in from a high to low concentration, and that provides energy to then move glucose with it. So both glucose and sodium are coming in. Now, as sodium comes in, though, it's going to go to equilibrium. So we have the sodium potassium pump down here that pumps that sodium that just came in. It transports it out using energy. So ATP is needed for that. But that keeps the concentration high outside, low inside to provide energy to move that glucose in. Now, once the glucose is in, we get facilitated diffusion of the glucose going from high to low concentrations into the blood, but it does need a transport protein to do that. So then it hops on that hepatic portal vein and then goes to the liver. And then we also have the lacteals, those lymphatic vessels there that would be absorbing nutrients as well. Now our lipids, so this includes triglycerides, which are fats, but also phospholipids, steroids like cholesterol, and some of our fat-soluble vitamins. So lipases are the enzymes that break down lipid molecules that release the free fatty acids. So we have lingual lipase, that's in the saliva, pancreatic, released from the pancreas, and gastric, released there in the stomach. So this allows for emulsification, which is when those bile salts turn the large liquid or lipid droplets into smaller ones. So it's just breaking them down into easier to manage molecules. So then the surface area is increased so the lipase can actually get to them better whenever they're broken down, instead of having this big bubble of lipids that are all kind of hidden, breaking them into individual smaller circles of lipids makes it easier for those enzymes to get to them. And those droplets of lipids are called micelles. So if you think about pouring oil into water and it forms the little droplets, those would be micelles. So those get absorbed by epithelial cells, and then they get repackaged into what are called chylomicrons, because again, they don't like water, so we can't be transporting them. So they form chylomicrons that enter those lacteals, the part there of the lymph system that's absorbing these chylomicrons into the lymph and then ultimately into the cardiovascular. So if we look at the transport of lipids here, so these bile salts are going to surround the fatty acids to emulsify them. So it's going to form a micelle. So it's just lipids surrounded by the bile salts to make them more soluble. So then that micelle is going to be endocytosed. So it's going to be taken into that apical membrane, into the intestinal cell. Then we have our triglycerides that are released. Then this protein coat is going to form what's called a chylomicron. So proteins essentially are just going to coat around these triglycerides. Then exocytosis is going to be released as a big molecule, like a big circle. And those are going to go, though, into the lymph. So these lacteals of our lymph system absorb those chylomicrons. A few different lipoproteins that are used to transport these lipids because, again, they don't like water. So these lipids have to be carried by a combination of these proteins and phospholipids that form lipoproteins. So cholesterol, about 15% is ingested, but most of it is made by the liver, and that was one of the functions of the liver, is to make cholesterol. So chylomicrons, as we just mentioned, they're mainly lipids. So those get formed by those intestinal cells, and then they're picked up by the lymph system. Then we have VLDLs, very low density lipoproteins. This is about 92% lipids, 8% protein. This forms around lipids that leave the liver, and then those triglycerides are removed from the VLDL and stored in adipose tissue. And then ultimately, some gets converted to LDL. So LDL is low density lipoprotein. It's about 75% lipid, 25% protein. This transports cholesterol to our cells. And cells have these receptors that allow them to take that cholesterol in. So the number of those receptors becomes less as our lipid cholesterol needs are met. So they're not producing as many, which means they're getting stored. So we actually want this LDL level to be low. This is kind of storing, packaging, and again, storing cholesterol in the body. Whereas HDL, high density lipoprotein, again, about half and half of lipid and protein, so high density. So many more proteins, but this is going to transport excess cholesterol for removal. So we want to have high amounts of HDL to help export that excess cholesterol so it doesn't get stored and we don't get atherosclerosis and the buildup of that cholesterol. So LDL stores it, HDL helps excrete it. They're both carrying cholesterol, helping to transport this molecule that doesn't like water. They're just doing different things with it. And this is just looking at what these look like. So again, we have on the inside, so the chylomicrons and the VLDL. Those have the triglycerides in there and some cholesterol, but again, mainly the triglycerides inside of them, surrounded by this phospholipid layer here. So again, the fatty acids like the lipids. The hydrophilic head here likes the water, and then we have some different proteins that get embedded. So chylomicrons don't have a lot of protein. The VLDL, we are adding a few more proteins in here, but again, transporting mainly fats from the liver. Then our LDL and HDL, this is mainly moving cholesterol, but again, cholesterol with LDL is being stored, HDL is exporting it, and the HDL has the highest amount of protein to lipid ratio. So if we look at the transport of the LDL as an example, so we have this low density lipoprotein, so it binds to the LDL receptors. That forms a pit to form endocytosis, which is where it comes in the vesicle to bring that LDL into the cell. All right, now moving on to our proteins. So we're going to start in the stomach with pepsin. So again, there's no protein digestion in the saliva. It starts in the stomach with pepsin that breaks it down into smaller chains. So if we think about a long amino acid protein here, what's going to happen is pepsin is going to come along and just kind of break it into individual pieces, into these little peptide, polypeptide regions. So pepsin is helping to break it down into more manageable sizes. Then the pancreatic protease like trypsin and chymotrypsin carboxypeptidase, they're going to help produce more of these smaller chains. Then peptidases on that brush border, they're going to result in like dipeptides, just like 2 amino acids or 3 amino acids. So again, just continuing making them shorter and shorter. And then those get transported into the epithelium depending on the type of amino acid. So some of them are acidic, or the neutral ones can be co-transported with sodium, like we looked at with glucose. The basic ones enter by facilitated diffusion. So there's a transport protein that they can attach to bring them in. So their energy isn't needed for them. Then after absorption, the amino acid goes through that hepatic portal vein to the liver and then ultimately into the cardiovascular system. So if we look at this amino acid transport, so again, we have an individual amino acid that was from these, the breakdown of the protein. Again, we get the symporter, so sodium comes in, going from a high to low concentration that provides energy to pump the amino acid in. It can then leave by facilitated diffusion, just that transport protein into the cardiovascular system, into the capillary. So very similar to what we'd seen with glucose. And then again, that sodium potassium pump is going to pump the sodium back out to keep that gradient existing to allow more transport of more amino acids. So then if we kind of summarize the digestion here, so again, starting in the mouth, we have that lipase and the amylase, so that's going to help break down our starches into disaccharides or monosaccharides. Then in the stomach, we also have the pepsin that's going to help break down the amino acids or break down the proteins into shorter chains. The pancreas releases that pancreatic amylase, just like in the saliva, to break down into smaller disaccharides or monosaccharides. Pancreatic also releases lipases to break down our lipids, as well as things like the chymotrypsin and trypsin to break down proteins. And then on the our absorptive cells of our small intestine, again, we are absorbing and secreting different enzymes here as well. So like the disaccharadases get released by the intestine, some lipases get released there as well, like the bile salts that come in. And then we get the pepsin that helps with the breakdown of the proteins. And then water also is going to get reabsorbed here, so it can move either in either direction. So it can be coming out of going into the GI tract or being reabsorbed following those osmotic gradients, meaning it goes with the solute. So if there's a lot of sodium and chloride and other ions inside the cell, water comes in to dilute them. If there's a lot in the digestive system, it comes out of the cells. So it goes with that concentration gradient of where there's more solute. There's also the oral rehydration therapy, where water contains sodium and glucose, and so water again flows there by osmosis and allowing it to be regulated. So again, with our water, we talk about the different ions, so particularly sodium, potassium, calcium, magnesium, and phosphate. These ions are actively transported. So if they come in our diet, in our different, especially things like Gatorade, but also different fruits and vegetables, they're actively transported, meaning they need energy to come in. Chloride ions can go by passively in the small intestine. They just go with sodium, but then they're actively transported in the ileum. And then calcium transport depends on vitamin D, and it's actually under hormonal control of parathyroid hormone and calcitonin. They control the absorption of calcium. Now, what happens with aging on our digestive system? So as we age, The mucus layer starts declining, so less mucus, a little bit less fluidity moving through the digestive system, less connective tissue, muscles and secretion. So ultimately less digestion. The flow through the digestive system decreases. There's increased susceptibility to infection because, again, we're not moving things through. They're hanging out longer. Those immune reflexes are decreasing as well. More chance of ulceration because, again, as stuff stays in the stomach longer, those acids start breaking down the intestinal layers. GERD, which is acid reflux, again, as that acid can then flow back into the esophagus. Cancers can form there as well. The enamel on the teeth and gingivitis chances also increase. And then just finally wrapping up this, talking about a common, so we mentioned vomiting as a reflex and of course defecation, but then diarrhea, which is any change in the bowel habits where we have increased frequency or fluidity of our stool. So acute is kind of self-limiting, just meaning, you know, it comes and goes. It doesn't last very long. Chronic is defined as lasting for more than 2 to 3 weeks. So this results from a decrease in fluid absorption. So remember, the large intestine absorbs about 80 to 90% of the water. However, diarrhea is whenever there's not as much water being reabsorbed and it's released in the feces. So mucus secretions by the large intestines increase dramatically and has large amounts of that bicarbonate. So the hydrogen ions remain in circulation, and that can cause metabolic acidosis. So it causes the pH in our system to go up because, again, so as the water moves out through the diarrhea, so does those bicarbonate ions. So some causes of diarrhea include an infection like bacteria or viruses, parasites, or also some various toxins. And the treatments are just replacing the lost fluids and ions, again, putting that base in. So like Pepto-Bismol, bismuth subsalicate, and loperamide to just help decrease the acidity of the stomach to bring things back to normal.